how do we see, or what do we see? Lines. Everything that we see is lines. Okay? It's all a series of lines. So each column, I just drew this up here to help remember. Each column in the visual cortex, each column responds to a line of a particular orientation. Okay? So if the line is at a particular angle, we'll show you where these angles come from. If the line is at a particular angle, that one cell fires. When that cell fires, more information about that line is sent to your brain. If you only see that one cell firing, what would you experience? If I only fire these basic simple cells here, what would you experience? Not a red. From end to end, as far as visual perspective is, a line of a particular orientation. So if I had my magical electric cord and I was sticking it and zapped inside the brain, you would experience a line, a sensation, going straight across. So that's where we have to get to today. So because this is so irritating. I'm looking at the board back there, and that's staring at you. Okay, I'm looking at the board back there because we don't want you to think I'm creeping on you. Okay, so if I'm looking at the board back there and I turn my head sideways, did I just change? If I turn my head like this, did I change where that board is going into my eyes? Yeah, my eyes aren't rotating with it. So if I change my head angle, that vertical line is still activating the vertical line cell in my brain. So what your occipital lobe is seeing is not actually what's there. It's a context of what's there. So like when you look at the ceiling, which way do these lines go? Well, if I'm actually like this, it's at a diagonal from here and it goes that way. Are these lights the same width here as they are over there? But they don't look like they're the same width. I know because of how I understand the space that these lines are parallel to each other. They might not be parallel to each other, but why do they look parallel to each other? My occipital lobe is organizing what I'm seeing into meaningful lines. Your occipital lobe organizes what we're seeing. So we have to get from your eyeball back to the occipital lobe here. It's actually kind of tricky. It's not that easy. Because as we go, I have a cool little graphic here that I made a couple years ago. Okay? How we get back, can you slide back and hit the lights off so we get the wheelie chair? There you go. Excellent. Okay. So, as we look at these, let's make a move this full screen. Yay. Okay. So what do we call these different parts of the eye? This is actually a napkin here at the lower right here. So we look tiny. How do you look at your eye? Okay. What is the black dot called? Pupil. What does it do? Right, so you actually, the pupil is the part where you're actually seeing back into the eye. What color does the pupil turn when you shine a light on it? I'm so happy you did this. What happens when you shine a light on it? What color is the pupil? Red. Well, yeah, because when you flash, if the light flashes quickly, what do you see? The retina. The retina is red. What color is the dog's retina? Some dogs are silver, some dogs are green. What about kitty cat? Cats are generally orangish. Okay, and then if you see like a deer or a bunny rabbit, they're silver. There's these little obnoxious birds that they're called them trail showbirds, but I think they're actually the gnatcatchers. They sit in the middle of open spaces in the middle of the night. And what they do is they look up. Good morning. Okay? When these little birds sit on the ground and look up, as you're riding your bike toward them, their eyes glow orange, the same color like a cigarette butt is on the ground. And your brain goes, oh, that's fire, that shouldn't be there. And right when your brain sees that, because they're looking up, they fly straight up right in front of you. And so that happens a lot at night, especially out here if you're riding a mountain bike at night with headlights. You see these stupid little glowing embers, and it looks like, ooh, is that a cigarette butt? Why would someone have a cigarette? And all of a sudden, bird flies up in your face. Terrifying. Okay. But, so when you see the back of the eye, okay, this is what we're looking at. We look at the back of the eye. Here is the pupil, and then the iris, the colored area, opens and closes. So as you're sitting in the dark right now, what is your iris doing? It's getting bigger or smaller? It's getting bigger. Because what it's doing is allowing more light to come in. And where's my pen? And the more light that comes in, copy. I'll get more efficient with this as we go. Oh, how come we didn't paste? Oh well. Okay, so one of these days I'll figure this out. Okay, so as the light changes the side, it changes how much on the back of the eye the light is focused. Okay, so if we have our pen here, okay, the pen is projected on the eye. Does it matter if it's upside down? No, because what your eye is seeing is the relative pattern of lines that this is. It doesn't matter which way it's oriented, because your eye is taking this information, and this information is being coded into these visual receptors in the back of the eye. Please notice how big the optic nerve is. Remember we showed you pictures of my mother-in-law, how big the optic nerve is? It's literally in your brain as big around as this pen. Huge amounts of optic nerve tissue coming out of your eyeball. So there's a lot of information coming out of the back of your eye. And when we go through how it works, you're going to realize that the information has already been filtered before it gets to the optic nerve. So these are really crazy things, how your eyeballs actually get information to the brain. Tremendous amount of information is coming back. You're not just seeing something once, it's being activated multiple ways in your brain at the same time. Okay, so get into it. So now, the center part is called the fovea. When we were talking about the glasses, these are kind of fun because what they do, they just focus in a little bit tighter. These are good old-fashioned reading glasses. And they focus the light so that I can see things up close. Okay. Oh, not really close. Okay, so why do I still look different with my glasses on and off? Wait, we thought we just said detail, line. Line, because we're talking about line. So if I have my glasses on, I can hold it up pretty close. Or if I do something with what's cool with my camera, with my cool camera here, hold on, I can take a picture and zoom in on my little praying mantis friend that I caught yesterday. Okay, so now by zooming in, what can I see better? Finer and finer and smaller lines. And that's what's really cool about the camera lenses on our new phones. Look at your new iPhones, which is impressive. I don't have a Samsung, I'm just saying. Samsung has got the best camera. I see the Huawei phone has the best camera to track your anyway, there's a chip in the Huawei phone that keeps track of who you are and where you are. They don't tell you that. Hey Google, are you listening to what I'm saying? Yeah. Yeah, so Oh, I turned the sound off. But it's always listening. Not going to be anything. Okay. So, but okay. So, but look at the wind. The more you zoom in, what do you get to see? The more detail. And the more detail is the more the ability to see the difference between one line and the line next to it. Okay? The ability to see two lines means detail. Without my reading glasses on, I have a really hard time reading the font that's on this computer. I keep the resolution high so that I can show pictures like this, but the Photoshop.com, I can't see that when I'm sitting here. I can make out the Google, but I can't see the rest of it. But when I put my lenses on, what can I read? I can differentiate the different lines. So in the center of the eye, around the fovea, we have high detail. We have very high amount of detail. And we'll explain why in just a few minutes. But that's why if you need corrective lenses, it's important to wear them because you focus your brain, you focus the information on this high detail area. What happens when it's dark though? Is it easy to see in the dark? Not necessarily only the rods, but so we're going to explain why that is in just a few minutes. So we'll talk about rods and cones in a second. But if it's dark out, my light is spread over a wider surface on the back of your eye. When it's bright light, the eye is focused on the center part. Then you try to read a book in the sun. Seems like a great idea. You got plenty of light, but then what happens? You get blinded. You actually can't differentiate the lines because what ends up happening is the light is so bright, all the cells get stimulated, and you lose the ability to differentiate lines. There's actually something called snow blindness. Okay, you don't get that much in San Diego, just saying. But when you go up, if anybody's ever been up in the mountains and gone skiing or something, sometimes the fog rolls in. And when the fog rolls in and it's snowing out and everything is covered with snow, what happens? You can't see. So we always ski with polarized lenses on our glasses. Polarized lenses cuts out a certain frequency of light, twice laterally or vertically, depending on the polarization. And so by cutting the polarization, by cutting the lens down, what does it do for the amount of light in your retina? Notice how it's not equal across the entire field. Notice how it's different colors, kind of looks like stripes on. Depending on how I move it, what do you notice? That's because it only allows certain frequencies of light to come through, so you cut out some of the lines coming through. It's really important because polarized lenses allow you to see lines better. It also reduces glare, and I think it's better for your eyes. I'll ask the person who works at an eye shop, but I knew you'd be able to answer.
Okay, one more question. So you know how it's like that while the glasses are on, when it takes them off, it will make your eyesight like it's not possible. It's not going to make your eyesight easier. It's going to prevent the vision problem that you're having with how it's confusing the eyesight. Because when you have the glasses and doing the theory, it's not just medicine where it's going to work better. It's just like a piece of plastic or a piece of that you work through. And it does do it for you. It's like having an app that's doing the work for you or something. Yeah, in front of your eyes.
So a good way to think about it is if you're riding a bike, your feet don't hurt. Because you're pushing on the pedals and it's not high impact. The ground is being touched by the wheels. So you're still doing the work, and it's a tool that helps us use less muscles so that we can be more effective. But it's not, the eyeball muscles they do get stronger and weaker, but it's not something, they don't fatigue that fast. Yeah, but with readers, this is really, I've noticed this a lot. The more that I wear them, the more that I need them. Yeah. No, it's not. I'm perfectly flexible okay. I'm working on my denial campaign of aging. Okay. I'm not getting older. Okay. So when we look at the eye, there's actually a blind spot where the nerve. So the back of the eye is called the macula. This is the shiny surface with the macula on the back of your eye. When we see things, light passes through photoreceptors to the macula reflects it back toward the center of the eye. So the light passes back and forth twice across every photoreceptor. It goes through on one way and then comes back on the other way. There's a disorder called macular degeneration. And macular degeneration is usually caused by sunburning eyeballs. So this is why wearing your sunglasses, especially with the UV protection. You go outside and you get sunblock, you put sunblock on your skin so you don't get a sunburn, but it's hard to put sunblock in your eyeballs. So that's why wearing those UV protecting lenses is so important, because it keeps the back of your eye from getting sunburned. My son has really sensitive sinuses, so he's having a hard time finding pair of sunglasses that he can wear. So he can wear it. So when we go fishing, it's hard for him to wear sunglasses because they hurt. They're like pressing on his sinuses back here. So we've been trying to find different pairs. I have a pair of old Ray-Bans that he actually likes that he can wear that don't hurt his head. Because when you're on the water, the light shines off the water, it shines back into your eyes, and the light's coming down in your eyes. So even if you're not looking at the sun, it's still shining in your eyes. Yeah, and that's why you get sunburned so nicely down by the water. Anyway, so the macula is really important because the layers of the rods and cones are for the center of the eye. Okay, so we won't go into too many eyeball disorders and things, but again, I showed you this picture before. That's where the optic nerve comes in from the eyeball. Okay, so move the back. Okay. These are the actual pictures of the columns and layers inside the optical nerve. This is better than my drawing. So we have the basic simple cells, primary visual cortex, area 1. What happens as we go up higher? Technically this is upside down, but that's not important. What do you notice about the density of the cells as the layers get more molecular? Well, they get, let me put this way. So here's the simple cells right here. So this would be like starting at the bottom and going up toward the surface of the brain right up here. Here's at the bottom of the brain. What do you notice about the density of the different layers? They get more dense the more complex the line is. We'll explain that again in a few minutes as well. But as the cells get more complex, we see the higher density of neurons. In other words, more neurons are communicating in order to form the more complicated lines. Simple lines are just a particular frequency. Complicated lines are more and more cells interacting. Okay. I don't know what there. There we go. Okay. Okay. Before we get into logical codes, I want to talk about two things. First of all, this answers your question. Not the question you're about to ask, the one you asked before. How do the lines get into the cortex? Okay, so what was your question next? Yeah. It's so frustrating. Yeah. Right, somebody posted a picture in the developmental class of the gray, the teal and gray bands, which are actually white and pink, or the pink and white ones under a teal light. And it's so frustrating because once you look at it long enough, you can see it's pink and white. But if you don't, you see it as gray and teal. Yeah. Let's get to that in a few slides and then answer that question yourself. Okay, so depending on the color blindness would be the answer, but okay. So now, this is something that in this idea of what's called a receptive field is really kind of hard to process in I think at this level of this course. I didn't understand them truly until I took an advanced neurobiology course where the professor studied eyeballs and studied how receptive field works, and he explained it for like 3 weeks, and then I finally got it. So basically, a receptive field is a cluster of cells. It's a cluster of cells. So it's a cluster of these photoreceptors. We'll go how the photoreceptors work here after we talk about the receptive field. But the photoreceptors basically sense light. That's how. So if I have an area of cells that are sensitive to light, this one receptive field sends 2 signals. This one receptive field sends 2 signals. This entire receptive field isn't very big, like as big as this is. The 2 signals it sends are off, center, on, outside. So if I shine light on this, what it's going to do is it's going to say, oh, here's the outside, here's the inside. What does this one do? It's the exact opposite, where the inside, when I shine a light anywhere from one side of this to the other side of it, the outside stops sending signals, and the inside starts sending signals. So, what about this little one right here? On the blink up here. So if I shine a light anywhere in this circle right here, it says send signals on this neuron, stop sending signals on that neuron. Anywhere within this receptive field, the off center says shine on the outside, don't shine on the inside. So each receptive field has 2 ganglion cells that come out of it. Yeah. Okay, so the big one and small one are the same idea, but why does, sorry, being male, why does size matter? The amount of information it can take in. So if I shine any point of light, if I shine one dot of light, there's actually something called a photon laser. It sounds cool, but it really isn't. So if I aim it at your eyeball and I shoot it, you can perceive a single photon of light. If I nail a photoreceptor right away, you see a quick little flash of light. Pretty cool. If I stop one photoreceptor from firing, you see a little flash of light. If I shine that little focus of light here, you see this whole area lights up. So that little point of light could be here, could be here, could be there, could be there. It doesn't matter within this receptive field, that area lights up. So this is, if we were actually to draw the eyeball, we do this on this one. So if I draw the back of the eye, okay, so let me draw the fovea right here. I'm going to draw receptive fields. These are all little what are these? Well, we'll talk about the photoreceptor in a second. Yes, they're cones. But so if I shine a light right here, right, I can tell the difference of this receptive field because it sends a signal, and this receptive field doesn't send a signal. So that's a pretty tiny distance apart. So when she used the word periphery, the receptive fields out here are very big. And in fact, in some places in your eye, they're huge. Why would you want huge receptive fields way out here? The larger they are, the less information you take in, but the more likely they are to fire. Okay, so I'm using information a little bit differently than you are. Right? So that's so awesome for this. So thank you for. If I shine in one little pin of light here, you see a pinpoint of light because it activates one receptive field. If I see a pinpoint of light here, the whole area lights up. The whole area lights up. So these are what we call magnocellular pathways. Oh. They're called magnocellular pathways because they're big areas. Magna means big. Okay, these little guys in here are called parvocellular pathways. I just remembered we had this here. So I got it. Okay. Parvocellular pathways are little tiny receptive fields. You haven't got to a line yet. Yeah. So, yeah, flies are really neat because each eyeball is a receptive field. So a fly has like a bazillion eyeballs, and each eyeball itself is its own little receptive field. So humans have all the receptive fields clustered on the back of ours. Again, if you want to take the neurobiology course, and you can start learning about how different organisms. My favorite are the slugs. Light or dark, that's all they get. In other words, somebody moved my rock, it's daytime, it's nighttime. So when it's light out, the slug goes, ha! And hides. When it's dark out, the slug walks around. If you move the guy's rock, it freaks out because it goes from dark to light very quickly. So that's like the most simple form of photorecepting eyeballs. And the slugs are all down the side of the back. Because that's what's important to it. If the shadow goes over it, it hides. My son's been trying to catch, when we go fishing, tries to catch crabs, the big crabs that walk around the rocks. You can never catch them because their photoreceptors, they look up. Because if something's going to eat it, it's going to come from above, a seagull or something. So they're very good at dodging things coming from above. They're terrible at dodging things that come from the bottom. So you take a plastic bag or a little piece of fishing net, and you put it on the ground, put a dead thing on it, they walk right on top of it, and you pick them up. And you're like, oh, surprise. You try to reach from the top. Yeah, lobsters are pretty stupid to begin with, but if you try to reach and grab it at the top, they're very tasty. I think most tasty animals are relatively stupid. Except octopi are very tasty, but they're super smart. I'm very torn with octopi. Okay, sorry. So these magnocellular pathways, what that means, magna means large. These are very large receptive fields. They're in the periphery of your eye. So if I go back this way. Not very efficient. So here, the receptive fields are very large. So any sort of light hits there, that cell fires. But there's no detail out there. Because a light can be almost a centimeter left to right, and you can't exactly tell it's left to right 1 centimeter back and forth. But in the fovea, you can see things as fine and apart as human hair. You can, if you have the right lenses on, or a magnifying glass, you can actually see crazy amounts of detail, especially in bright light. Okay, and so especially like anybody have one of the fancy new TVs? The Ultra HD something something TV? Yeah, we go to Costco and we just stare at them. I don't see well enough to own one of those. I don't need one. Because the TV can produce signals that are stronger than the human eye can actually see. You get to 4K, you're good. That's about the max resolution that an eyeball has. Unless you're playing video games, then it's important to have the Ultra HD LED 5, whatever it is now that's fancy. Okay, so getting back to the main point. Okay, blink again. I don't think it, oh, it does. Yay, okay. So I forgot that I had done that, and then okay. So why did I have on-center and off-center cells? Remember, I say cells because the cell, there's hundreds or thousands of photoreceptors within the receptive field, but what do they talk to? Ganglion cells. They're in between the two. We'll draw that diagram out in a second. Well, it is, and that's why I remember when I first taught this class here, I just finished taking the advanced neurobiology course at San Diego State. So I was trying to explain exactly how photoreceptors work and all the different biochemical processes and all this, and you guys are all like, I forget. And it's a lot. It's too much. Because your job is not to become neuroscientists. This class is to teach, well, this class is to find the neuroscientists and make them ask all these really good questions and make them go and dig into it more because it's so freaking cool. But again, we keep struggling with that as a department. We keep talking about that too. It's like, how much detail do you really need to know? Is learning the big picture and the basic concept important, or are all the knowing details important? Well, in like statistics, sadly, a lot of the knowing details are what's the important part to learn. Because you gotta know what it says, period. But in this class, understanding the big picture allows you to ask the questions, which allows you to find the details. That's why we start at the visual cortex first, and we're slowly working our way into the way the eyeball actually sends signals. Because these are the ganglion cells. The ganglion cells are the ones that actually produce the optic nerve. So the optic nerve, again, this becomes irritating. The optic nerve is made of these ganglion cells. So this is what is getting sent to the brain. Okay, on center, off center. Every pathway has ganglion cells. So I'm going to draw. Let's see, how could I draw this? I'm going to draw some white dots in here. So, in the parvocellular pathway, some receptive fields are on-center, some receptive fields are off-center. So some send signals when the periphery is lit up, other ones only send signals where the center is lit up. Not phobia. The center of the receptive field is lit up. So I'm going to go back here again. So some receptive fields activate on the edge, some receptive fields activate on the center. Sometimes they're overlapping each other. So you have lots of receptive fields as layers of ganglion cells have different receptive fields that read the information coming out of the eyeball. So far, so good? So two receptive fields like I drew here, could they overlap each other? Yeah, and so this is a yellow and a blue receptive field. And when yellow and blue receptive fields overlap each other, they make green. So if a wavelength shines here, Your brain sees, oh, yep, this yellow firing blue, yellow firing blue firing. But to answer your question about corrective lenses, I can, if you are red-green colorblind, the most common form of colorblind, if you're red-green colorblind, anything that's red or green looks relatively the same. You still see it, but you can't tell red or green. It looks the same. And you ask the person, is it red or green? They're looking at you like, I don't know what red and green are, so yes. My good friend John Ross is completely 100% red-green colorblind. Cannot see red, cannot see green at all. He's just missing that gene, yeah. Well, of course, because where's the red light? On top, and the green light's on the bottom. Doesn't matter. So he did have trouble in, is it Amsterdam, or the traffic lights go this way. It only takes one, though. You figure it out pretty quick. What's red? Yeah, so it's perceived light of a particular wavelength. He can't tell you what is red, what is green. I'll play a video for a minute. I play an intro class it does follow the male chromosome much more than the girl. Sorry. So that's why they have the colors written on the labels too. But again, so if when you look at something that's white versus pink, how can you tell the pinkness of it versus the whiteness of it? Based on the density of the light coming in and the amount of these red photoreceptors being fired in your eyeball. And if you don't have the red photoreceptors, what comes out of the pink? The white. It's just like trying to decide between navy blue and black in darker light. Yeah, it's impossible because navy blue is so close to black that unless you have enough bright light, you cannot tell the difference. Or if you're under some store lights, have you ever gotten something home and the store lights put out a different wavelength? And then the colors look really weird? We ended up painting a wall at our house. We were trying to go for a khaki, nice brown color, and we ended up getting a skin color. It's like, ugh. We just painted our living room. This is back when we were first married and we didn't know what we were doing. So we actually just bought a gallon and painted the wall before we even let it dry and looked at the colors. Learned that quickly. Yeah. Color is very subjective. So even if I wear, what color are these lenses? It's kind of a brown color. But I can look through brown, I like amber lenses, they're my favorite. I can look through brown lenses, and does it change the color of things that I'm looking at? Only for about 1 or 2 seconds. But after a period of time, what happens? Your eyes don't adjust. Your brain adjusts. So when you ask the question is, can I wear corrective lenses to change the frequency, to change the color blindness, you can wear lenses that change the color spectrum. And if you wore a lens to change the color spectrum, it could take the pink shirt and push it into a spectrum that the person would be able to identify. Would they see it as pink? Maybe. Depending on their learning. But again, I'll play that video here in a bit. Okay, so in the magnocellular pathways, you also have overlapping on-center and off-center. Overlapping on-center and off-center. The reason why they overlap on-center and off-center is because your job is to differentiate lines. Okay, so I'm going to draw a line. I'm going to zoom in right on the photoreceptors here, and I'm going to draw a line in magical sparkles. How do I see that line? Shining a magically sparkling line across the center of your fovea. Don't say photoreceptor, say ganglion cell. Ganglion cell. We haven't even got to photoreceptors yet. So this ganglion cell says, I'm firing, but not in center, or just on center. So this tells me that because there's on center and off center ones, it decides where the edge of the line is very quickly. Okay, we'll get there in 2 minutes. So then, does this cell fire? Does that one fire? No. Does this one fire? Yes. Does this other one fire? If it touches the edge, does it fire? Yes. So your brain forms a pattern of receptive fields. If the pattern of receptive fields are at a particular angle to each other, if this one is at a particular angle to this one and that one's at a particular angle to this one, that photoreceptor, those ganglion cells go to the thalamus then goes to the occipital lobe, and that occipital lobe cell at that angle says, ew, me! Right? Because of the relative angle of the cells to each other. Please don't ask how that works. I don't know. But it gets, okay, go ahead. You would have spaces where a light could hit and not trigger one or the other. You don't, and that's actually when you have damage to your retina or macular degeneration, that's what happens. You have the macula absorbs the light. I'm looking over to confirm that I'm saying it right. But when you have damage to the macula, my grandfather had macular degeneration. He lost his foveal vision because he was out on he was a Navy guy. He was out a pilot flying above the clouds without good sunglasses and on the ocean all the time without good sunglasses, and he was roasted his eyeballs. So he didn't have much fovea because the light would go in and it'd be absorbed by the non-shiny substance in the back of his eye. And if it's absorbed, but the macula is actually just, it gets, the photoreceptors go, I'm not seeing it. So he has a gray spot wherever he tries to look. He can still see peripherally. We actually had to take his car keys away because we caught him driving. Why did he keep doing this in order to drive? So he's looking around the gray spot. He's driving using his magnocellular pathways. Don't do that. Because, yes, you can stay on the road, but what do you miss? The details. And so his little golf cart is the last thing that we finally had to take away. They have little posts that you have to drive through before you get on the golf cart paths in Rancho Bernardo to keep cars from driving on the golf cart path. He ran into those posts so many times that homeowners are like, you're going to get kicked out of living here if you don't stop driving. And he kept bashing up his golf cart because he can see the path. He can stay on the path by doing this. He cannot see the posts. Well, if he turns his head and moves his head like this, he could see the post. Because the post, if he's moving, so let's get back to moving then. What's moving? So we'll make a nice sparkly line over here. So you would activate, if I activate these 3 receptor fields, you see a line going about that direction. How come we can't tell exactly what the line looks like out here? So the line could look like that, the line could look like this, the line could look like that. Are those all activating those 3 receptor fields in the right sequence? Yeah, so you see a big fuzzy line over here. You know it's a line, but I don't got much details because the receptor fields are firing. But what's cool is if something's moving, okay, like this little thing right here, how do I know it's moving? One receptor cell becomes activated, the next cell becomes activated, and this one stops being activated. This one's activated, the next one's not activated. So if a cell's activated and not, a cell's activated and not, a cell's activated and not, in a particular direction, your brain in the magnocellular pathway says something's moving. Something's moving. So when you see movement out of your peripheral vision, yes, that's the magnocellular pathway. There's a really cool disease, well not cool, but it's called motion blindness. Motion blindness is where you lose the part of the brain in the thalamus that senses the magnocellular pathway. So you can look around and everything looks perfectly fine, but you can't see things moving. Yeah, so why would you not be able to see something moving? They are. Your brain can't hear them because the part of your brain that recognizes the sequence of firing is damaged. So you look at something and all of a sudden it's gone. Well, why is it all of a sudden gone? It's there and then gone and then appears somewhere else. Because you can't see the sequence of movement because that's the magnocellular pathway. There's an old video somewhere on my computer of a guy who has motion blindness. They are flashing things to him and he can read the words. But then, when it's flashing, it's fine. When it stops and slowly starts to move, I can't see that. Because it's moving and the part of his brain that... So we have two parts of our brain now. We have the occipital lobe, which is seeing the lines, but we also have another part of the brain that the thalamus is talking to, and the thalamus is talking to the middle temporal cortex. And the middle temporal cortex is right in front of it, and that is giving you the movement. So the lines mix with the movement, then start giving us shape and form and activation of more. Okay. Very rare. That's why they talked about it in the cool video. There's another really cool one called the Capra delusion, where the person, I just thought it was a good idea. Okay, where the person, you guys can actually watch that lecture because I might be gone on Thursday, and I can send that lecture out to you. Sorry, it's about the delusions and all the different weird things. Okay, no. Okay, moving on. So, if the movement areas are out here, the lines are in here. Okay. You have, sorry, I'm going. The whole video is coming back to me, and I really want you to watch it now. Okay, so how do the cells actually fire? Let's go in there. Okay. Let's go into actually how the cells fire. Oops, I don't know what that. Oh, that's the wrong one. Sorry. I'm sorry, I got completely sidetracked, but I recorded it, so I'm more of an idiot. Okay. So let's zoom in. This is the light shining in from the bottom. So this would be, if I were to do this properly, I would turn it this way. My eyeballs are oriented this way. And so if I take this, hold on. How come you're not pasting? It should, if I hit Alt print screen, it should print. And then I should be able to paste it. So if I hit copy that, I should be able to go to here and hit paste. There you go. We'll turn it properly. There we go. So like the eyeball was, now we have this nice little image. So light is shining. Light is shining from this is the eye, my pen is over here, my pen is being shown on the macro, on the back of the eye. Okay, the photoreceptors are right along the edge of the macula. If the macula loses its shininess, the light cannot shine back onto the photoreceptor, and you lose about 50% of your vision. What's backwards? Because they get more light. If you put them out here, light would shine and bounce back, but in the distance that it's shown and bounced back, it would take time. Yeah. So light shines through the cells, and on its way through, it hits, this is a cone, and that's a rod, that's a cone, because the cones are cone shaped. Okay, the rods are rod shaped. Okay? So as the light shines through, it hits the macula, and right as it hits the macula and bounces back, it hits the density of the photoreceptor. So the idea is that you want to try to get the light to fire the photoreceptors as close as possible. You don't want the be way out here, because the light would shine through, bounce off, and then lose some more energy shining back through. So it's really funny that I taught this for years. It never dawned on me that the photoreceptors were close to the macula until like 5 years ago. It just never dawned on me that, why? Because it's just, we just saw the diagram, memorized the diagram, and just went on with it. So that's why I really like teaching this stuff. I love this stuff. I would suggest a fly has the receptive fields on the surface of their eye, but they don't really have much lens on their eye. So I think my guess would be that most animals with a lens that focuses light tend to do this. It concentrates the ability to see light. So organisms that are like bush babies and organisms with really big eyes have really shiny maculas, because they can absorb very small amounts of light and focus small amounts of light. Like those little bush babies, mort. Oh, I opened SPSS by accident. Uh-oh. Those guys. So, why do they have such ridiculous duality in animals? You don't know, man. You don't know what I see. Yeah, I mean, like, you can just don't feed them at night. Okay. Yeah, that's so. No, they are cute. Okay. Okay. Yeah, it does definitely look a little strung out, I must say. But the reason why these guys look like that and see like that. Sorry, SPSS, I didn't mean to open it. I keep getting excited. You're gonna open me? No. Okay. So the reason why they see is that they can see details at night. In almost pitch black, they can see details. And they can also see color at night. There's another pathway that I didn't draw on our brain, which is called cellular. These guys, the little guys look at you can tell if fruit is ripe at night without having to climb up and get it. Because as a little creature that with not many defenses, your best effort is stealth. And so by being able to look up at the tree and see which critter, where's the critters and which fruit is ripe to eat, I can get there faster and back much safer. So for humans, though, that would be a really cool thing for you to look up. To try to figure out that answer to that question. Why? Well, what biological adaptation is immaculate? My answer would probably be detailed vision. My guess would be that organisms with, I know the purpose of cones. Oops, so magical. The purpose of cones is detailed vision. Because cones, if I shape something like a cone, I can put a lot of them in a very small area. And I can differentiate between 1, 2, or 3 cones in a very, very tiny space. So in the fovea, most of the receptive fields are tiny and made out of cones. Most of them. There are rods in the phobia, but the rods are pretty much just sensitive to any light. The cones, however, are specific. They're sensitive to certain wavelengths of light, and this is where color vision comes from. Some cones have a propensity to fire more for yellow, some for blue, some for red, and some for green. Or is it green and red? I can't remember exactly which one. Green, blue, yellow, yeah. Trichromatic, yeah. Red is the absence of green. What happens when you mix red and green together? Yeah, they cancel each other out as far as the light goes. If I shine a red light and a green light together, it turns into black. They're opposite each other. Okay. Okay, so let me zoom in a little bit more. So again, this is a little irritating, but I'm going to write this in the dark. A rod is constantly firing. In the dark, rods and cones are constantly firing. You see in the dark. No, if it's completely dark, cave blindness, the opposite of snow blindness. You cannot see. If someone, well, it happens with coal miners every so often. Every so often a coal mine or a deep mine, they will have some sort of accident. And nowadays they make them carry these little hand crank light sources. Because if they're stuck under the ground for a couple days, what can happen? They actually go blind. Because of lack of light stimulation, their cells stop firing. You need your cells to be firing in order for you to see. So wait a second. So you don't see darkness, except in your soul. But you don't see darkness. What do you actually see? Okay, so so now this is kind of fun. Let me put this, I'm going to line here. Let's turn on the lights. Yeah. Okay, so can you just reach back and flick them both on real quick. What happened right as they came on? It got darker and then it got light again. Okay, so keep back there again. Wait for it. Okay, shut them off. You see it get brighter and then it gets darker again? Okay, one more time, then we'll leave them on. See it gets darker and then it gets light again? I know. You don't really notice that until you start doing that. Yeah, but when you walk outside and it's super bright out, you can't see, everything is too dark. Yeah, but when you can't see anything, everything is too gray. You can't differentiate where anything is. It's actually too bright in there, in your head. You have to wait for a second for your eyeballs to settle down a bit. In the digital theater, which is really fun, I keep in there. It is so freaking dark in there if you keep the lights off. It's because it's made to be very dark in there if you set the lights off. And it's really crazy when you first walk in. If I've done this multiple times, where I go and shut the door, I have everything shut off. I go shut the door and I turn around and realize, crap! I have to walk all the way back across to try to get to where the podium is. And if you don't have any sort of semblance of space, as soon as you take one or two steps, what happens? You can't see anything. There's no orientation. You can lose your orientation very quickly because where you are has to do with the nitrocellular pathways and movement keeps everything moving around you. Like if you close your eyes and you stay around a bunch of times, all of a sudden you have a really hard time understanding where exactly you are. But as soon as somebody makes a noise, or as soon as you feel the board, that's actually your hippocampus. Like, oh, I know where you are, and build the world around you. That's how a blind person can navigate actually pretty well, because they build the world around them. But they build it by other things. Our neighbors have a blind dog, and it does just fine. It does great. You can't tell it's a blind dog. But every so often, what does it do? Runs into things because it can't see. But it learned to walk slowly because hurts less when you bump into things. Yeah. Yeah. Yeah, it's like when we put in our deck on the back of our house. We took the ghost step many times. There's the 2 steps going off the back of our house, and you can't really see them. Now we've had the deck there for almost 15 years now. We don't take the ghost step anymore. But before, we used to walk on the deck, you just, boom, in space. Okay, so the way this works, okay, I'm just going to go through a nutshell because I don't want to run out of time. Want this in your head. This releases glycine. In the dark, iPSCs are being sent, glycine, to the bipolar cell. So, in the dark, I did this demonstration before, but in the dark, the photoreceptors are constantly sending iPSCs to the bipolar cell. Bipolar cells are not manic or depressed. What they just are connection. Okay, connection? Sure. They're just a direct connection cell. And the direct connection cell just goes back and forth between the rods and the cones and the ganglia cell. Okay? So, the rod and cone is constantly firing in the dark. It's constantly sending inhibitory signals. As it's sending inhibitory signal, what does the bipolar cell do? I'm going to change back to the right. In the dark, what's a bipolar cell doing? Nothing. It's going to constantly send signals because all cells do. So it's going to fire really slowly. However, as soon as I turn on the light, though, what's going to happen? Why is it going to fire a whole bunch? Slow down there, Nikki. Okay, so in the dark, the bipolar cells do this constantly inhibited. It's still going to fire a little bit because all cells fire. But as soon as I turn on the light, the cone stops firing. What stops being inhibited? Bipolar cell. And when the bipolar cell stops being inhibited, it sends signals. It sends an excitatory action potential to the ganglion cell, which increases the ganglion cell firing. So the ganglion cell fires in the light. You see light. But the photoreceptors are set to fire when it's dark. No, rods and cones. Rods and cones. Notice that they only go back and forth. The rods and cones to ganglion cells. So how can you see some other cells that will go out and discuss it and go back and forth laterally? That's where the lines come in. That's where the photoreceptors come in. The receptive fields come in with the lateral cells. The ganglion cell fires when I turn off the rod. If I turn the rod off, it's actually a pretty simple process. I won't go into detail because it's not as relevant. But when I hit a photon to a chemical in the rod, it exposes sodium channels. That's it. Is it immediate or is it slow? Slow. You can actually see it. It takes long enough for you to physically perceive how long it takes to slow down. You can see it. You can see the world changing. Exactly. What's fascinating is that with the brighter the light, because we're just talking light and dark here, very, very simple. But what about looking at the periodic table? What is that? How do you perceive the periodic table? Lines and colors and shapes and patterns and textures and it's paper, it's wrinkled, it's shiny. There's a lot of light information coming off there, and our eyeball is taking that entire light and dark sequence and mapping it into the back of our eye. Simultaneously all the time. So when I use the word light and dark, it's a relative term. How do I know what is light and what is dark? So I just drew this as an on-off. Right? It doesn't work that way. It'd be nice if it was just on-off. But so, here, can you just put the lights down again? Both of them. That's on you. Same time. There you go. Wait. Go on. He's back. One more time. There you go. Okay, so I want you to look at the color of the board up here. Okay, watch the color of the board. Okay? So if you just stare right below ECSP at the board. Okay, now put them off. Do you notice it actually changes? It's not just once, but it's actually whoop. So stare at that center of the board every ECSP and put it back on. Do you see your peripheral change faster than the foveal? So because the peripheral, they can reset a lot faster because there's a ton more receptors in the periphery than there are in the fovea. In the fovea, all those little receptors have to reset it, refocus, regenerate. Okay, so usually that happens because your irises keep opening and closing and opening and closing. Okay, especially if you're hungover, no offense. But when you're hungover, it takes longer for your cells to reset. And so your muscles in your eyes, most of the headaches caused from looking at stuff are caused from the muscles in your face trying to move too much. And they just get fatigued, which makes you strain more, which makes them fatigued, and it causes headaches. A lot of times, like migraines also. Migraines are when you have the nerves that go to feel your face get overstimulated. Sometimes your eyes can't. Sometimes the best thing with migraine is get in the dark and close your eyes. Well, it is because any stimulation coming in the facial nerve can make that migraine feel worse. Because a lot of people's migraine is blood flow generally, and by just opening up your eyes and looking at more things, more blood flow flows to the area, and it just hurts more. Migraines are awful. Okay, most migraines have a trigger. If you can figure out your trigger, you can get rid of your migraine. Yeah, it's like Frank, he gets really bad migraines. His is weather. He can tell you when a Santa Ana's brewing because he gets really bad migraines. Lacey, it's wheat. Her son has gotten really sick as a baby, found out he has bad celiac, so they did the genetic test. Her husband and the other kid are fine. She is very bad celiac. Never knew it because she doesn't have the intestinal problems. But she doesn't digest certain proteins in wheat, which cause bad migraines. She stopped eating wheat, and all of a sudden she hasn't had a migraine. She had a cookie at Disneyland, because you have to have a cookie at Disneyland. Migraine. Oh, totally worth the $18 for the cookie. We're actually going to, right after the Halloween stuff, when the Christmas comes out, we always make a road trip up there and get the gingerbread cookies. You don't have to go into Disneyland, but the big gingerbread cookies, highly recommend. Okay, they're my wife's favorites. We get it for Christmas stuff. Okay, so last thing in a few minutes, in order to make a receptive field, okay, we have right here 2 receptive fields. Why do we have 2? Oh, sorry, 1 receptive field. 1 receptive field. We have 2 ganglion cells going on. Okay? 2 receptive, one receptive field, 2 ganglion cells going on. If light shines anywhere on these 3 neurons, what activates? I'm sorry. two, these two. The receptive field fires. Remember, these two cells, the ganglion cells, are the receptive field. Okay, this is oversimplified, but this is how it works. If I send a signal and a light hits either one of these two, if it's an on center, this one fires, this one doesn't. If it's an off center, this one fires, and that one doesn't. Okay? So there's this little horizontal cell right next to it. If light is shining on this receptive field, this ganglion cell fires. If light shines on this receptive field, brain cell, this one tells these not to fire. Okay? Two receptive fields right next to each other. Why would they tell each other to fire and not fire side to side? Well, I can show you what it looks like. I want the big. They used to just put the big images up here. Now that I'll make the whole thing. There you go. Perfect. Okay, hit the light stop again. Okay, so just look right at the center. What do you start to see in the corner? Dots start showing up. You just stare at the center, all of a sudden this little cluster becomes darker. And the longer you stare at it, what happens? The darker they get. So if you look at this box up in the corner up here, you stare at the box up in the corner up here, you'll start to see little darker squares show up in between the other squares on the side and the middle. And if you look at the bottom corner, you'll have different patterns show up. Very irritating. Okay? Because if you look at it on a computer monitor with higher resolution, it's even more frustrating. There's another one. We should have another one here with the dots on it. It's this one right here, but I don't know if it'll actually let me. That one's bad. Because are there really black dots? Yes. No, they're not really there. But why do you see them? No, your brain can figure it out. Your eyeballs can't. Okay, so what's happening is if you look at the center black box, this is a very complicated pattern across your retina. You're activating and deactivating a series of different receptive fields. Okay? So this is activating an on center, this is activating an off center, this is activating a little bit of on center, a little bit of off center. And so when I look at the center, in my fovea, the receptive fields get too big. I can't differentiate the little dot. So sometimes it's on center, and sometimes it's off center. And literally, the little black dots blink. And it's so frustrating because if I'm up close like this, and I stare at it, they don't turn black, they turn gray. It's like blink. Oh boy, when I blink, it's really bad. But for me, every time I turn my eyes to look at it, why do they go away? Oh, we're over time. Why do they go